So the second presentation was about hyperprogressive disease. It was a session organized by AACR together with FDA, because several clinicians observed, not just in non-small cell lung cancer, but in other diseases, that in a percentage of patients, there is the sensation that the immunotherapy is able to increase the tumor growth. So the world is divided into believers and non-believers. The non-believers are people who think that this is just the representation of a more aggressive disease, and a group of people who are convinced that there is a group of patients in which you can have an acceleration of the tumor growth. So there was an interesting panel of people who worked behind this phenomenon. In our institute, we worked a lot behind this phenomenon, and we found a particular immune phenotype, which is an immunophenotype characterized by macrophages with an epithelioid morphology co-expressing PD-L1, CD33 and CD163, and expressed in all patients in which we think that this phenomenon can exist. So we are working on a prospective trial to test this phenomenon. On parallel, the preclinical laboratories of our institute reproduced the phenomenon or an increase of tumor growth in some animal models with the same genotype in which we observed the phenomenon in humans. And we think that behind the phenomenon, the innate immunity is involved, and there is a switching of the macrophages from N1 to M2 through the Fc portion of the immune checkpoint inhibitors. We are not totally sure about the mechanism. Also, the tumor can play a role with the PD-L1 expression on the tumor cells, but we are working on that. Anyway, in the panel, it was very important. It was very important because we thought that it was very important to to harmonize the criteria to define what a hyperprogressive disease is, because every group is calling hyperprogressive disease with different criteria.